Hello and welcome. My name is Lahcen Akerkouch. On behalf of myself and our team at the Department of Civil and Environmental Engineering at North Dakota State University, I'm presenting our work of high-fidelity simulation of flows in bone-like environment to investigate the growth of cancer cells. To create a 3D surface mesh of the scaffold, we use the micro-CT scans of the actual physical scaffold used in experimental studies in format of Lahcen files. We then import that to a open source software imaging like Osiris in this case, when we took multiple threshold regions to capture the maximum holes as much as possible, as shown in figure from 1 to 2. And from that, we try to grow the region so we can have 3D shape of the surface mesh as shown in figure 3. So from figure 3, we can see that the shape of the scaffold is highly complex and very hard to simulate and to resolve all the holes and porosities. So as a first step, we took the half of the scaffold to simulate. As shown in figure 4, we have the computational domain with a resolution of over 27 million grid points to resolve as much points or all of the points as much as possible. In figure 5, the velocity contour is shown in it. The results reveal that the flow at the vicinity and within the scaffold is highly complex and non-uniform. And to investigate the effect of pore sizes on the flow velocity, we took a cross-sectional plane of the scaffold as shown in figure 6, showing that the flow velocity is very sensitive to the pore sizes. In conclusion and future works, we did investigate the profile of fluid flow in the scaffold by performing a high-resolution CFD simulation on the full-scale 3D computation model of the scaffold based on the micro-CT scans. Those results showed the degree of complexity of the flow distribution inside the scaffold, and the method used for the simulation, the immersed boundary method, showed that it can tackle this kind of complexity with accurate results, which is very promising for future works when we plan to simulate the full scaffold and also multiple scaffold inside a bioreactor. Thank you.